Hello again, everyone. So thank you for continuing on through this session. Thank you, Sandro, for an excellent talk. I want to make one point of clarification in the logistics here. There have been some questions that have been raised for Sandro, and if you have more questions for his talk, continue to put those in the questions box at the lower left of your screen. And at the end of the next talk, we're going to have a joint question and answer session. Sandro will be live with me, and the two of us will answer questions. The next talk is actually from me, and so I am just a brief introduction to me, but I'm an assistant professor at Harvard Medical School in the Department of Microbiology, as well as in the program in Cellular and Molecular Medicine at Boston Children's Hospital. And as you'll see in this talk, my laboratory leverages multiplexed RNA imaging, which is a nice complement to the protein imaging that Sandro just discussed, to get at the same types of questions, the identity of cells and their spatial organization in tissues. And so I think this provides a nice bookend where you can now see how multiplexing has been improving in both the protein space and the RNA space. And so again, we'll have all our questions for both talks after this talk. Good afternoon, everyone. My name is Jeff Moffitt, and I just want to thank the entire organizers for this opportunity to be a part of this session. So I'm an assistant professor at the program in Cellular and Molecular Medicine at Boston Children's Hospital and in the Department of Microbiology at Harvard Medical School. And today I'm going to tell you a little bit about a core technology that I co-developed as a postdoctoral fellow, and which is a major driving force in my laboratory. This is a technology known as multiplexed error-robust fluorescence in situ hybridization, or MERFISH for short. And in a nutshell, it's an approach that allows us to image and identify hundreds, thousands, or now tens of thousands of different RNA molecules simultaneously within individual cells in fixed tissue samples. And because of that, we can now do spatially resolved single-cell transcriptomics and use these measurements to both discover and define cell types and states while mapping out their intricate spatial organization in intact tissues. So today I'm going to tell you a little bit about how this technology works, how it performs. I'll give you just a taste of the wide variety of biological applications that we are now tackling with this, and then I'll dive relatively deeply into one, I think, very evocative application, the use of this technique to discover and map cell types in a portion of a mouse brain. Before I go too far, though, I want to acknowledge that I'm a co-founder and stakeholder in a company that aims to commercialize the MERFISH technology. That company is known as VisGen, though no products or services of VisGen were used to produce any of the data or results that I'll present here today. So why would one want or need spatially resolved approaches to single-cell transcriptomics? Well, I'm sure you're all very well aware of the revolution that we've seen in our understanding of a wide variety of tissues, driven by our ability to dissociate, capture, and sequence individual cells using technologies such as single-cell RNA sequencing. So we can now take any complex tissue, dissociate it into individual cells, extract from those cells the RNAs they express, and then quantify their expression at the transcriptome scale using next-generation sequencing. And that in turn has allowed us to now discover and catalog cell types in almost every organism and tissue. But in the process of dissociating these cells and extracting these RNAs, we lose important insights. So we lose insight into how the RNAs are organized within the cell, and that organization can be a very potent form of post-transcriptional regulation that drives a wide variety of biological phenomena. In addition, we lose insight into how these cells are actually organized within the tissue, and as we know, tissue organization plays an intricate role in how individual cell types communicate with one another, and how the behavior of the tissue as a whole emerges from the behavior of the individual cell types and states that comprise it. And so what we wanted to do was to develop technology that had the strengths of single-cell RNA sequencing, but could preserve this spatial context. And that's what we've done with MERFISH. So MERFISH is based upon a very powerful technology known as single molecule fluorescence in situ hybridization, or FISH. In this approach, you fix your sample, permeabilize the membrane of the cells, and then hybridize to it a set of fluorescently labeled DNA oligonucleotides, whose sequences are complementary to different regions of the RNA of interest. This concentrates a large number of fluorophores in each and every molecule of that RNA, such that when you image this sample with an epifluorescence microscope, you now see bright fluorescent puncta for each molecule. This allows you to count the number of molecules per cell, and of course, see their organization within the cell and the organization of that cell within the context of the sample as a whole. And so our goal with MERFISH was to take this powerful technique and multiplex it. We do so by assigning to each RNA a unique binary barcode, and then we read out those barcodes in the following way. We stain our sample using fairly conventional single molecule FISH approaches, but with one important distinction. Instead of targeting a single RNA, we target every RNA that has a 1 in the first bit of the barcode. We get an image like this, we'll remove this signal, now restain the sample, targeting every RNA that has a 1 in the second bit of its barcode. We'll get a different pattern of fluorescence, which we will then image and extinguish, and we'll repeat this process for each bit in these barcodes, such that the fluorescence on-off pattern we see across the stack of images defines the barcode and thus the RNA. Now, one of the important features of this is that we also leverage barcoding schemes that allow us to detect and correct when individual errors can happen. So not only can we identify large number of RNAs via the large number of barcodes produced by this method, but we can do so with very high fidelity, as I'll show you here on a slide. So here's an example of an actual MERFISH measurement. What you see here on the left is an image of a human osteosarcoma cell culture after the first round of MERFISH imaging. Each bright fluorescent spot is a single RNA molecule. We can zoom in on one small portion of one of the 3 cells you see in this image, and you can see that one molecule is present, which gives us a 1 in the first bit. We remove this signal, restain the sample. Other molecules are now fluorescent, but this molecule is no longer fluorescent, giving us a 0 in the second bit. We do this again, in the third bit, this molecule is now fluorescent again, and I think at this point you get the idea. In this case, we'll do this 13 more times to build up a stack of 16 images. This molecule is on in the first, the third, the 8th, and the 13th image, giving us the barcode that you see here. We have this barcode, or similar barcodes, I should say, for all of the 140 different types of RNAs stained in the sample, allowing us to reconstruct an image like this, where each marker is an RNA and the color is its identity. And so you can see that this approach allows us to optically barcode and identify individual RNA molecules. And we've extensively benchmarked this technique. So we know that it now has a large dynamic range, near 100% molecular detection efficiency, and faithful detection even of a single molecular copy per cell. And by increasing or changing the length of these barcodes, we can tune the multiplexing to the demands of our specific question. We can identify hundreds, or thousands, or now 10,000 different mRNAs simultaneously within individual cells using MERFISH. And because this is an imaging technology, and imaging can be quite rapid, we can actually perform these measurements in tens, if not 100,000 cells in each measurement. So this can be a very high throughput technique as well. So with this ability to do massively multiplexed single molecule imaging within individual cells, we can now ask a pretty broad range of questions. For example, we can use the natural variation in gene expression from cell to cell to identify sets of genes that covary and thus are likely co-regulated. So we can identify or delineate regulatory modules. We, of course, know exactly where each and every one of those copies was found within the cell, so we can map the internal organization and transcriptome, and we can do this in a wide variety of cell types, states. We can use the fact that gene expression is a faithful measure of the state of a cell to infer properties of that cell, not just type, but state. We can also use this technology to measure synthetic RNAs, not just native RNAs. And these synthetic RNAs can serve as barcodes that allow us to identify individual cells. For example, cells that carry different genetic perturbations. And so this opens up a wide variety of exciting biotechnology applications as well. And then finally, we can use the gene expression measurements we have for individual cells to define their type and their state, and in many cases, discover unanticipated types and states, all while mapping their intricate organization in tissue. And it's this application that I'm going to primarily focus on for the remainder of my talk. So I'm going to tell you about a published application of MERFISH for discovering and mapping cell types and states. This application was from the mouse brain, in particular, a portion known as the preoptic region of the hypothalamus. This was work done in collaboration with Catherine Dillock while I was still a postdoc in Xiaowei Zhuang's laboratory. Now, what you see here on the left is a coronal and a sagittal slice in the mouse brain, and the preoptic region is the region in the black box, and approximately in the region of the dashed line. Now, this is an interesting brain region because it's responsible in part for the control of a wide variety of homeostatic functions. So it has circuits that control how a mouse responds to hunger, thirst, or fever. And it also controls higher level functions that are nonetheless non-learned. Social functions, for example, how a mouse responds to an aggressive perturbation or a situation that involves mating or parenting. We know through the work of a variety of labs that there are neurons in this region of the brain that comprise circuits that are activated and in part control these behaviors. But what wasn't clear was the diversity of neuronal types that were found within this region. And so we wanted to use MERFISH to ask the simple question: what are the different types of neurons in this portion of the brain, and how are they linked to these hypothalamic functions? But not just what neurons are there, but how are they spatially organized? How do they distribute into the already defined subregions in the hypothalamus that have been linked to a variety of different hypothalamic functions? And then can we go beyond just identifying these neurons based on their molecular profiles and also mapping them spatially to inferring their functional organization into circuits? Now, Murphish is well suited for all of these measurements, but because this was the first large-scale test of this technology for discovering and mapping cell types, we wanted to have an independent measure of the cell type diversity that was present there. So the first thing we did was dissect out and dissociate single cells from the preoptic region of multiple male and female mice, capture them using the 10x Chromium platform, and perform single-cell RNA sequencing. That produced the data that you see here on the left. This is a matrix in which individual rows represent different genes, columns are different cells, and color is the expression of those genes in those cells. We can then cluster these cells using standard statistical methods and identify those clusters based on the genes they express. And as expected, we find all of the major classes of cells one would anticipate in this portion of the brain. We can then visualize this high-dimensional data in 2 dimensions using a technology known as t-SNE, where each cell here in this 2D space, or each marker here in this 2D space is a cell, and it falls next to other cells that express the same genes. The colors are now indicated by the clustering of these cells. And we can then take out our inhibitory and excitatory neurons, marked here in red and yellow, subject them to a second round of clustering, and we found about 66 different types of neurons in this portion of the brain. We then, in parallel, performed MERFISH on exactly the same brain region, taken from different mice. We measured in total 155 different genes with this technique, taken from 3 different categories. First category were classically defined markers of major cell types. So genes that define inhibitory neurons, excitatory neurons, oligodendrocytes, and so forth. Second class of genes were inspired by what we discovered with our single-cell RNA sequencing and were included because they could discriminate different neuronal types. But we didn't want to just define neuronal types, we also wanted to learn something about their biology. And it's known from a wide variety of prior work that neuropeptide and hormone signaling is an important mode of communication in the preoptic region. And so we included in our panel a third category of genes, which was a comprehensive set of neuropeptides and neuropeptide and hormone receptors. And the receptors turn out to be an important group because these are genes that can be quite functionally relevant even when expressed at very low levels. And despite its many strengths, one of the weaknesses of single-cell RNA sequencing is that it captures a relatively modest fraction of what's actually expressed in the cell. And so it can be challenging to extract statistically robust measurements of these lowly expressed but functionally relevant genes. By contrast, MERFISH has single molecule sensitivity, so we hope we could do a better job here. What you see here on the left is a result from a MERFISH measurement. We took a 2 millimeter by 2 millimeter by 600 micron block out of the preoptic region, sectioned it into 10 micron thick sections, sampled every 5th section, arrayed those on cover slips, and measured them with MERFISH. What you see here on the left is a 200 by 200 micron field of view from one such slice. Each white dot is again a single RNA molecule. We would include with these measurements co-stains that allowed us to automatically define the boundaries of these cells. That's what's marked in white. And then we could color these RNAs based on their identity as determined by MERFISH. And I think you can immediately see from this image the transcriptional diversity that is present within just a small portion of this tissue. You have cells that are expressing predominantly red RNAs, green RNAs, blue RNAs, and so forth. These are a visual representation of the molecular fingerprints that define different cell types. Now, to give you a sense of scale over which we can perform these measurements, let's zoom back to a single 2 by 2 millimeter slice. That's what you see here. The white box is the field of view that I just showed you. And now we don't have a color palette that can accurately describe to our eye 155 different genes. So I'm going to simplify this by just showing you 8. And so we can walk through the spatial distribution of just 8 different genes collected in this single slice. And I think you can immediately see the richness in gene expression across these large tissue areas. And by leveraging the high throughput of MERFISH, we could in total characterize gene expression in 1.1 million cells with tissue collected from 31 different animals and a total integrated tissue area of over 700 millimeters squared. Because we have the boundaries of these cells and the RNAs that fall within those boundaries, we have an expression profile for each and every one of these cells. We can now analyze these using exactly the same statistical toolbox developed for single-cell RNA sequencing. So unsupervised clustering and t-SNE visualization. That's what you see here. And what we found was that 155 genes was more than sufficient to find all of the major classes of cells we expect to find in this brain, as you can see here. And now we could actually ask a very interesting question. We've now characterized gene expression in this region of the brain using a technology that dissociates and sequences cells, and one that images RNAs directly in intact tissue. Both technologies find the same classes of cells, but to what degree do their expression profiles agree? So we performed a pairwise Pearson correlation analysis. In this matrix, each row represents a cell type identified with MERFISH, each column the comparable type identified with sequencing, and the strong diagonal here indicates that these two technologies agree very nicely. The fact that these correlation coefficients on this diagonal are all very close to 1 means that this agreement is actually quantitative. So it's a nice cross-validation of both techniques. So MERFISH with 155 genes can define these cell types. The advantage, of course, is that we never dissociated the cell. So we get an atlas of this tissue essentially for free. That's what you see here. Each black box is a 2 by 2 millimeter slice. There are 12 slices taken from a single animal, and they are arrayed from the anterior to posterior portion of this brain, left to right. The gray boundaries mark individual cells. You can now color these cells based on the identities determined by the expression profiles as measured by MERFISH. So here are our inhibitory neurons, our excitatory neurons, mature myelinating oligodendrocytes, oligodendrocyte precursors, microglia, astrocytes, endothelial cells, mural cells, and ependymal cells. These are ciliated cells that drive the movement of cerebrospinal fluid, and you see that they form a single cell layer flanking a structure known as the third ventricle filled with that fluid. And so we can now put back all of these cell types to reveal the intricate organization of this portion of the brain from a single mouse. Now we can take our inhibitory and excitatory neurons as measured by MERFISH, subject them to a second round of unsupervised clustering. And in this case, we find 70 different types of neurons. We can then do the same type of Pearson correlation analysis with single cell sequencing that I presented a few slides ago. We see a nice strong diagonal, again, providing a nice validation of these two technologies. But we can go one step further and look to find a correspondence between the neuronal types we find in MERFISH and neuronal types we find in sequencing. And we did the simplest thing one could imagine, which is to pull out the largest and statistically distinguishable Pearson correlation coefficients. Those are the elements marked here in this matrix. Now, what's nice about this is that we find that the vast majority of the neuronal types that we see in MERFISH have a corresponding type in sequencing. That is a nice cross-validation again. But one thing I want you to notice is that this correspondence is not always one-to-one. There are instances in which MERFISH is able to subdivide a single neuronal type that sequencing identified. And what we find in most of those instances is that these subdivisions are defined by the expression of lowly expressed but functionally relevant receptors. And so the sensitivity of MERFISH allows us to discover cell types that we were incapable of discovering with the statistical approaches that we used with single cell sequencing. We can also take a look at the spatial distribution of these neurons. Here again, we see our 12 slices per animal arrayed left to right. Gray are all cells, and red, green, and blue mark 3 of the more abundant inhibitory neuronal types. I want you to see that these patterns are bilaterally symmetric and that they change in striking ways over relatively short distances along the anterior or posterior axis. These patterns are also nicely reproduced across replicate animals, and I don't want to go here into the details of what these spatial distributions mean. There's a lot that they tell us about the organization and the principles of organization of the hypothalamus. But what I want you to take from this is that if we look at 9 different types of the 70 types of neurons that we found, they all have their own unique spatial distributions. So there's a remarkable spatial richness in this small portion of the brain. Now I want to tell you about one last experiment we did in this work. What we did is we took a female mouse we added a pup to its cage. It turns out that that mouse will actually go and retrieve the pup, bring it back to its nest, and care for it, even if it's not its own pup. That's called the non-learned or instinctive parental behavior. After that behavior, we then sacrificed the mouse and performed MERFISH in its brain and included one extra gene in our panel. This was the gene CFOS, which is rapidly expressed in neurons after activity. We clustered and found all of our sending type of neurons and then looked to see which of these neurons were expressing CFOS. What you see here is a bar for each of our neurons, and the height of that bar indicates the enrichment in CFOS expression. And what we find is in these virgin female mice that have just expressed this parenting behavior, there's one and only one type of neuron with a statistically significant enrichment in CFOS. We can now do exactly the same experiment, but in male mice, and the results are strikingly different. Those mice will often kill those pups. What we find is they have a different set of neurons that are now activated. Now we can do a very interesting experiment. We can take a male mouse 3 weeks after it's sexually experienced. It will now care for those pups exactly like female mice will. And now what we see is that this parental neuron is activated in the background of an activation pattern reminiscent of the aggression. And so I think you can get the point here. We can now stimulate these mice with a variety of known stimuli that trigger hypothalamic functions and now systematically go through and functionally annotate the neurons that we've discovered. And one point I want to make here is that the neurons we find that discriminate aggression and parenting were not separately clustered in our sequencing data. These are defined by a handful of lowly expressed receptors, and we only see these neuronal types in our more sensitive MERFISH measurements. And so with that, I want to just leave you with a sense of MERFISH. MERFISH is a massively multiplexed single molecule imaging technique that functions by taking a series of images, all of the same sample, in which different subsets of RNAs are fluorescently labeled in each image, such that across the stack of images, each RNA has a unique fluorescence on-off pattern that serves as a barcode that identifies it. We can bootstrap this technology to the multiplexing of hundreds, thousands, 10,000 different mRNAs, so near transcriptome-scale measurements. We can do so with very high performance, near 100% detection efficiency, very low detection limits and large dynamic ranges, and we can quantify tens of millions of RNA copies in tens of thousands of cells per day at a cost of less than a fraction of a penny per cell. And we can use this technology to now create molecularly defined atlases of complex tissues. For example, in the hypothalamus, we created such an atlas in the preoptic region. We used this atlas to discover really a striking diversity of neuronal types in such a small portion of the brain, and to map them out, and to functionally annotate them. And the spatial organization that we observed has very interesting implications for both the circuits that are present in this region of the brain, as well as the developmental process that gives rise to them. The last point I want to leave you with is the fact that the technologies that we developed and used in the brain are not specific to the brain. And so that means that MERFISH can be used to create similar molecularly defined atlases in a wide range of tissues, which is exactly what my laboratory is now doing. So with that, I want to say thank you to all the people who are involved in this work. Mainly the work that I presented here, or all of the work that I presented here today, was derived from my postdoctoral time in Xiaowei Zhuang's laboratory. And the tissue work was all a close collaboration with Catherine DeLog's laboratory. We had a really great group of people working on these projects. Stephen Eichhorn, DJ Bambamuku, and Eric Vaughn were involved in the tissue work, and then Hao, Alistair, Stephen, Junji, Guiping, Tian, Hazen, and George were all heavily involved in different aspects of MERFISH development. And we had some excellent help in clustering from Karthik, Shekhar, and Aviv Rejev's lab. I also want to thank the members of my new laboratory who have been helping me get this technology established at Boston Children's and Harvard Medical School, and are now using this to tackle a wide range of questions. And I look forward to telling you about those results sometime in the future. So thank you very much for your time and attention. All right, so now we're going to open this up to questions. Again, if you have any questions that you haven't asked, there should be an option for you to add those questions to the list in the lower left of your window there. And so Sandra will be joining us here, I think, in a moment to answer a few questions. But in the meanwhile, I will go ahead and start with a MERFISH question. So we have a question from the audience that reads, does MERFISH require fresh brain? And can it work with frozen human brain? So the answer is that MERFISH is actually a very general technology. It can work with a wide variety of different preparation methods. We have worked with freshly frozen brain as well as heavily fixed brain, but it does require fixed tissues because you are repetitively imaging the sample, so you need to have molecules that are anchored in place. Great. So Sandra, there are a few questions here for you.
Great. Can you hear me, Jeff?
Yes, I can hear you now. Great. And so I'll just start at the bottom. Do you perform a strip and reprobe of tissues so you can obtain all the cell types in one single site? And then there's a nice compliment to say that the images are impressive, which I completely agree.
Thanks, Jeff. So the method, it's fairly straightforward. We incubate with antibodies. The standard way is 3 antibodies. And then after imaging, we photo bleach. So the antibodies remain attached to the tissue. And then we reapply an additional 3 antibodies and continue with cycles of application and then photo bleaching. So there's no stripping per se, although some methods that are similar to this one use stripping. We find that is a little more rough on the tissue and leads to tissue degradation. So we prefer the photo bleaching method. We are expanding from 3 to 4 antibodies now and out to 6 per cycle. So that's another way that we can increase the throughput of the assay.
Great. So Sandra, here's another question for you. This is a really good one. It's a question that actually applies to both technologies, but how does one go about analyzing these gigapixel multi-channel images? And, I'm going to addend that question. Can you just comment, a typical SciSIF measurement, what is the size of the raw images that you collect here?
Yeah, so we do whole slide imaging, so there's a lot of data. There can be in one slide there could be up to 1 million plus cells. So in one of the 3-dimensional reconstructions we showed, that data set there is a couple of terabytes of data. So it's quite large, and I'm sure we don't maybe we don't rival you, Jeff, but it's a lot. It's just a lot of information. So how do we go about analyzing these images? I think you can think about this in 2 broad ways. There's commercial software that's available. Companies like Halo, Indica Labs, Halo, Visiopharm, they have software that's available. Because we're really into the nuts and bolts of this and developing the methods kind of the way Jeff is with his technology, we really make all of the pipelines customized. So if you're interested, you can find these pipelines on GitHub. Most of it's written using MATLAB essentially. And you can think of it broadly in terms of pre-processing, where we have to stitch together these images and do artifact correction using a program called Ashlar and another one called BASIC. Then we do segmentation. Generally, there are a whole range of ways of doing this. Again, it's a whole important scientific area of discovery, but we use a machine learning-based bioimage analysis tool called Elastic, which is really great. We generate segmentation masks, then do single-cell calls of fluorescence intensity, and then it goes on. I mean, it just goes on and on and on from there in terms of cell type calling, neighborhood analyses. There's really a lot to it, and we can go on indefinitely. But I invite you to look at our GitHub, the Santagata Lab GitHub, the Lab of Systems Pharmacology GitHub with Peter Sorger, and also a few of our papers in eLife, Nature Protocols, and Nature Cell Biology, which will get you a good view of how this works. But Jeff, I mean, this for us, this is like our main thing. I think the key thing is the underestimation in terms of the amount of computational time and effort that's required. The acquisition is really rather straightforward in comparison.
Yeah, I would completely agree that what is exciting about these extremely multiplexed images is the amount of information you get from them, but you have to computationally extract that. Yeah. It's a non-trivial problem. It's not solved. And I think it is a place that needs a lot of active research. a place where innovations could make a huge difference.
Absolutely. Jeff, should I ask you a question from the list?
Sure.
So there's one that says, is there any limit for low abundancy transcription transcripts? Sorry.
Yeah, so in Murphish, there in practice is no limit. We have, in every measurement, we include a series of barcodes that could identify RNAs, yet we don't. So those serve as an internal control that measures our false positive rate. And that will vary slightly from measurement to measurement, but it's consistently at a level of less than a fraction of a transcript per cell. So that means that if you count a single copy of a transcript, you can actually trust that measurement. And so, this false positive rate does affect our ability to infer average expression that's much less than 1 copy per cell. But for most single cell analyses, that's not really the level that you're being able to extract statistically robust or interesting information.
And how about related to that, there's a question about RNA isoforms and then the ability to detect small RNAs, microRNAs.
Yeah, that's also an excellent question. And so the way that single molecule FISH in general performs is you will tile an RNA with multiple orthogonal probes, each targeting a different region. That's actually very powerful because it means that if any individual probe does not bind because there's occluding protein or secondary structure in the RNA, well, then you still have the other probes that bind, you get a robust signal. And it's one of the reasons why we can detect and perform so well in a variety of samples. But it also places a limit on the length of RNA. And so we can go quite short, a couple hundred nucleotides, but things where you could really only fit on one or a couple probes are going to be challenging for our current implementation of these techniques, though we're working on this.
Okay. Hi, Jeff, I think it's time for us to wrap up.
All right, I'm seeing that as well. Well, everyone, it looks like we are out of time, and there are some additional sessions that I think I encourage you all to attend. It would be excellent. And again, I want to thank Sandro for joining us here today and for the organizers for putting together what's really a phenomenal set of talks. So thank you, everybody.
Yes, thank you. Bye-bye.